What comes to your mind when you think of gene editing? For most people, it's this picture on this left. We have this new technology that's going to cut cancer out of our genome just like a regular pair of scissors. However, in reality, it's not that simple. I work in a zebrafish lab, and our goal is to make gene editing more efficient. You might have seen zebrafish in your local pet shop. However, they are also a fantastic model for scientific research. Unlike goldfish, they're very hardy and can live up to 4 years, and they also reproduce in very high numbers. And these resulting embryos are transparent, so we can easily visualize all aspects of development. So, in order to edit DNA, as shown here in 2 black parallel lines, we use a system called CRISPR-Cas9, which is composed of 2 key components. The first one, as shown in blue, is the Cas9 protein, which acts like a pair of scissors in that it can cut DNA. Along with this, we have the single guide RNA, shown here in red. This guide RNA acts like a GPS, telling the Cas9 where it should cut in the DNA. Once the DNA is cut, the repair mechanisms to fix this often make mistakes. So therefore, we have deletions at the site of interest, as shown here with the dotted lines at the bottom. In my lab, we attached an exonuclease, shown here in orange, which acts like an eraser attached to the Cas9 scissors. So not only does the DNA get cut, but it also erases part of it to make larger deletions, as shown with the larger dotted lines. We tested this in zebrafish embryos and remarkably not only found that we had larger deletions with exo-Cas9, up to 439 base pairs, than traditional Cas9, which produces about 10 base pair deletions on average, but we also had a higher rate of targeting as well. Now we can use this technology to knock out any gene in our body, even potential cancer-causing ones. If we make a small deletion in a potential cancer-causing gene, it might not be enough to completely knock out its function. But however, if we have a larger deletion, it's much more likely to completely knock out this function. We have shown that exo-Cas9 not only produces these larger deletions that could potentially knock out function at an even higher level, it also has a higher rate of targeting. And it is through these small innovations like this, and potentially even others, that hopefully within my lifetime, we can have the dream of curing cancer with gene editing become a reality. Thank you.